Today we are going through the immunity for International Pearson Edexcel specification. Here is the plan with the timestamps for today's lesson. Feel free to stop the video several times, as it is filled with huge amounts of required theory. Just make sure that you understand each part of the video before getting into the next part. At the end, you will be subjected with past paper questions from Pearson Edexcel specification. So we are going to begin with the basic introduction of types of white blood cells, or how are they also called, leukocytes. There are two major groups that mainly differ in structure of their cells, granulocytes and agranulocytes. How the name suggests, granulocytes will contain the granules inside of their cytoplasm when we will look at them under the microscope, while agranulocytes will not. There is one more difference. Granulocytes have lobed nuclei, which helps them to squeeze through the capillary walls during non-specific response, while agranulocytes have simple, mostly circular nuclei. There are two types of leukocytes from each group that perform phagocytosis, so they engulf the pathogen and digest it. So the first one is monocyte, which transforms into the macrophage once they come out of the blood into the surrounding tissue, and neutrophils. However, the macrophages, the ones on the right, can renew its lysozymes, which are hydrolytic enzymes inside of the lysosomes, so our vesicles with hydrolytic enzyme that are called lysozymes, which digest the pathogen. On the other hand, neutrophils, the ones on the left, they cannot really reproduce their lysozymes, so will last shorter, while macrophages will last longer and also do one thing we are going to talk about later, become an antigen-presenting cell, which is required for specific response. So next on the list is eosinophils. They are responsible for parasite combat and allergic reactions, and basophils. They will release histamine molecules that will be useful during non-specific response. And the last group, lymphocytes. Those are your T and B cells. We will have the separate segment of video dedicated to their analysis. But in general, they are responsible for the specific response. Okay, so now we're going to talk about the cell regulation. You need to know that each cell has its own molecules on their membranes that enable them to be identified. They are called antigens. They are mostly derivatives of proteins, glycoproteins, and carbohydrates. You also need to know that the things such as flagella in bacterium or some viral components may also be the antigens that may be detected by the immune cells of our body. Your immune cells recognize those antigens as self, so body cells, or non-cells. So in most of the cases, the non-self antigens are from the pathogens. So basically they are molecules on the membrane of the pathogen, such as bacteria or virus. Once something was recognized as non-self, so basically the complementary receptors on the immune cells have bound to the pathogenic antigen, the whole component that had this antigen, so in most of the cases the pathogen itself, will be digested by the phagocytes about we have already talked about, so macrophages or neutrophils. In order to digest the thing, they should firstly engulf the antigenic component or the whole pathogen. This process is called phagocytosis, where the engulfing cell ends up having the pathogenic components inside of the vesicle, so including our pathogenic non-self antigens. We are going to talk about what happens to those isolated antigens in the next slide. But basically, the macrophages is going to become an antigen-presenting cell, which will be useful during the specific response. Here is the overview of things that may happen after the pathogen enters the body. So basically, non-specific response will happen anyways in each case. It will cause inflammation in the affected region where the pathogen entered the tissue. This will also result in the increase in the local temperature and several other things. But most importantly, the phagocytes, so our macrophages and neutrophils, will try to digest the small quantities of the pathogen. In a scenario if the digestion will not play out, so when our pathogens will divide faster than they are engulfed, the specific response would be triggered. Specific response is specific to each type of pathogen, because if the antigens present on the pathogen will differ, we would require other T and B cells with other complementary receptors that may detect those non-self antigens, so the whole pathogen. There are two types of specific response. so our humoral response, and cell-mediated. We will talk about each of them later on. I forgot to mention an interesting thing that will also take place only if your cell got infected by the virus. Interferon signaling molecules are going to be released. We will also talk about it later. However, you should keep in mind the small map of the responses that may take place. Okay, so now we'll start diving deeper, so pay attention. We are going to talk about non-specific response. Imagine your skin was cut, which unveiled the underlying tissue. Once some pathogens will enter, it will be detected by the mast cells, which with basophils both will release histamine molecules. They will cause the capillaries under the attacked region to become leaky and the gaps between the capillary cells to become larger. So simply, increase the permeability of the capillaries. This will cause a bigger inflow of blood into that area. So you will see the redness in that area. This is the first stage, inflammation. Then you will feel fever in that region. The local temperature is going to rise over 37 degrees Celsius in that area. The reason for this is because most of the pathogenic bacteria will not survive in the temperatures over that threshold of 37 degrees Celsius. As the permeability of the capillary walls increased, the macrophages and neutrophils will be able to squeeze into the infected tissue from the capillary. And the next thing that you already know will happen, so phagocytosis. Neutrophils will digest smaller fraction of the pathogen, because their lysozymes in the lysosomes are not renewable, while macrophages will engulf roughly 70% of all that nasty things. Now we will talk about the action of the macrophages during phagocytosis. They can do one more thing that neutrophils can't. They will become an antigen-presenting cell. So basically, as our macrophage will engulf the pathogen, it will form the phagosome, the vesicle with the pathogen inside of the phagocyte. Then, the lysosome with the lysozymes will fuse with that sac and the pathogen will be digested. So basically, as our macrophage will engulf the pathogen, it will form the phagosome, the vesicle with the pathogen inside of the phagocyte. Then, the lysosome with lysozymes will fuse with that sac and the pathogen will be digested, leaving the things that made him detectable, so his antigens. Now pay attention. The macrophages will also transcribe the gene for intermembrane protein called MHC. Then, its mRNA will be translated and the MHC itself would be formed. Now the interesting part. The antigens that were left will bind with the MHC complex and the whole thing is going to be placed on the membrane of the macrophage. So now we have a macrophage with a non-self pathogenic antigen sticking out of it. You are going to understand why we would need it later on, when we will talk about T helper activation stage of humoral response. There is a group of proteins called cytokines. They are signaling molecules and they help lymphocytes, so our T and B cells, to be activated and also may be used as the chemical sign for pathogenic activities. After the phagocytosis happens in macrophages, they will secrete those cytokines which will allow more phagocytes and lymphocytes to come into the site, therefore reinforcing both specific and non-specific response. And one more thing left for non-specific response. Let's say that a viral pathogen entered the normal cell. So our normal cell got infected with a virus. In this situation, interferons, this type of cytokines, will be released from the infected cell to the neighboring cells. Those interferons will bind onto the specific receptors on the neighboring cells and will activate the cascade which will prevent the viral transcription and therefore viral replication. So in general, we will basically shut down the transcription and translation of the neighboring cells, so that our virus will not be dispersed around the body furthermore. Before we get into the humoral response analysis, we need to talk about the types of lymphocytes, so our T and B cells. Both T and B cells have their naive cells, so T naive and B naive, which will turn into the T and B effectors, respectively. So in the situation if T naive will be activated, it will transform into the T helper, so also called T effector, and our B naive, if will be activated, it will be transformed into B effector, so B plasma cell. T naive will become activated once the antigen non-self, which was presented from the APC, so our antigen presenting cell, for example, it could be the pathogen itself or the macrophage, which engulfed the pathogen, digested it, and placed the antigen on its membrane with MHC. It will bind to the T naive cell, T cell receptors, which are complementary to a specific pathogen. Once our T naive cell will be activated, it will undergo clonal selection, so basically divide by mitosis several times. This process will form two types of T cells, T helper and T memory cells. T helper cells, or T effector as we said, will release interleukins, so type of cytokines, to activate B naive cells. They also will release cytokines to activate T killer cells. So it will release interleukins during humoral response to activate B naive cells, and it will release cytokines to activate T killer cells during cell-mediated response. Let's talk about the memory cells. So both T and B cells contain memory cells types. So memory cell will basically be activated upon its binding to the antigen, non-self pathogen from the pathogen. It could be once more as a macrophage, or it could be the pathogen itself. And it will be transformed into the T or B effector respectively. So if T memory cell will be activated, so it will bind to the antigen, it will become T helper cell. And B memory cell, once it will bind to the antigen, it will form B plasma cell. Okay, so now let's talk about the B cells. B cells have a specific receptors, so basically receptors which can recognize the antigen. So our B cells have a specific receptors which are actually antibodies. You're going to see it later basically. When B naive will become an antigen presenting cell after engulfing the pathogen, this process would be involved in the activation of the B naive cell. Then our B naive cell will undergo clonal selection and produce B plasma and B memory cells. B plasma cells, or B effectors, will produce antibodies. They will simply synthesize more antibodies and release it out of the cell. And the same thing will happen basically with B memory cells as with the T memory cells. Once they will bind to the specific pathogen, so their antigen, they will be converted into the B effector or B plasma cell. So now we are going to talk about the specific response, specifically about the humoral response. For convenience, I created a table with specific stages that take place. Note that before T helper activation stage, the non-specific response takes place, specifically the macrophage action of phagocytosis that we have already talked about. At the bottom, we can see that all the lymphocytes, white blood cells, originate from the bone marrow. In order to mature, they need to divide by mitosis and translate their self-antigens and receptors that would be complementary to the green non-self antigen. So the first thing that should happen is the macrophage engulfing the pathogen and forming phagosome. Later on, the lysozymes in the lysosome are going to digest the pathogen, and the only thing left in the macrophage will be the pathogenic antigens, so the green stuff. Those antigens are going to be bound with MHC complex that we have already talked about, and our macrophage will become an APC, so antigen presenting cell. After that, our macrophage will travel to the lymph, where it will activate the T-naive cell with the receptors that may bind to the specific pathogenic antigen that were bound onto MHC on the membrane of macrophage. However, to activate T-naive cells, one more thing should happen. Our macrophage, after it binds to the T-naive cell with the MHC complex, it should also release cytokines, which will trigger the clonal selection of the now activated T-naive cell to produce T helper, so T effector, and T memory cells. This will result in production of sufficient amount of T cells with receptors complementary to the pathogenic antigen. If the T memory cell will bind to the pathogenic antigen, it will become active and transform into the T effector, so T helper cell again. Those T helper cells will then continue on to the next stage, humoral response. We need to discuss on how B-naive cells will be transformed into the B plasma cells, so B effector cells. Matured B-naive cell with receptors that enable them to undergo phagocytosis with the specific pathogen will engulf the pathogen itself, and by the same process as the macrophages, they will become an antigen-presenting cell. So basically they will digest it and place its antigens on the MHC. In order to activate B-naive cells to become B plasma cells or B memory cells, the T helper produced in the previous stage should bind to the pathogen antigen on the MHC complex of the B naive cell. The T helper cells will also release interleukins, which will bind to the B naive cell in order to activate it and trigger the clonal selection. This process will form both B plasma cells, so B effector cells, and B memory cells. And once more, once B memory cell will meet the pathogen antigen, it will transform into the B plasma cell. And now the thing that we were all waiting for. B plasma cells that were harvested from the previous stages will produce antibodies which are going to be released outside of the cell to fight the pathogen. Okay, so now let's talk about the specific cell-mediated response. Imagine the pathogen entered the normal cell, so our cells get infected. Not only the non-specific part of that thing will happen, so basically our interferons are going to be released to the neighboring cells to reduce the transcription and translation of viral proteins, but also the specific response will take place. Our infected cell will digest some portion of the virus or bacteria that came into its cytoplasm and will basically become an antigen-presenting cell. Then, T killer cell with the specific receptor will bind to that infected cell, and in order for the T killer activation to proceed, we also need the release of cytokines from the T helper cells. So we have a requirement of two processes to take place. So first of all, activation of T killer by the release of the cytokines from T helper cells, and second thing that should take place is for our killer cell to bind to the infected cell's MHC with the pathogenic non-self antigen. Once our process will be activated, our T killer cell will be activated, it releases enzymes to digest the infected cell. So two types of enzymes will be released. Hydrolytic enzymes, which will basically digest the infected cell, and perfoporans. They are basically membrane proteins which will create the ion channels on the infected cells. This will lead to the dysregulation of ionic balance, and our infected cell will die, so apoptosis will happen. B cells will secrete a lot of antigen-specific antibodies into the blood. Antibodies will bind onto the surface of the pathogen to form lots of antigen-antibody complexes. Antibodies are made out of 4 polypeptide chains, so they are a quaternary protein, and also they have a constant and variable regions on those 4 polypeptide chains. Constant regions are the same in most antibodies, and the only thing that changes is the variable region. The active site of the variable region is complementary to the specific pathogen, while constant region allows the antibody to bind to the immune system cells, such as macrophages or B cells. Okay, so now let's talk about the modes of action. Antibodies can bind to the toxins produced by the pathogens. This prevents the toxins from affecting human cells, so the toxins are neutralized, inactivated. Second type of thing that may take place is agglutination. Each antibody has 2 binding sites, so an antibody can bind to the 2 pathogens at the same time. The pathogens become clumped together, phagocytes then bind to the antibodies and phagocytose a lot of pathogens all at once. And the last thing, opsonization. Basically, antibodies will bind to the pathogen and mark them for destruction. Also, opsonization will prevent pathogens from binding to the human cells, because we're basically gonna block the receptors on the pathogen itself. Okay, so now let's talk about the primary and secondary immune response. Before the person is exposed to the antigen, there are none of the right complementary antibodies in their bloodstream. This is because the T and B cells haven't come into contact with the antigen. Shortly after the person is exposed to the antigen, the concentration of the complementary antibody begins to rise. This is the primary response, and it's slow because there aren't many B cells that can make the antibody needed to bind to the antigen. The concentration of an antibody peaks at about 20 days after the first exposure and then begins to fall. It begins to fall because the person's immune system is starting to overcome the infection. When the person is exposed to the same pathogen again, at, for example, day 60, the secondary response happens. The concentration of antibody rises quickly from the moment of the secondary exposure to a peak at about 7 days after the second exposure. This is because the memory cells that were created after the first exposure quickly divide into plasma cells. They will produce the right complementary antibody to the antigen almost immediately. The memory cells also recognize the antigen and divide into the correct type of T cells to kill the cell carrying the antigen. Now we will talk about the different types of immunity. There are two major types, active and passive immunity. Active immunity is when your immune system creates its own memory cells and antibodies. It will give you a long-time protection due to the formation of the memory cells. However, it takes some time for that type of immunity to produce sufficient amount of antibodies that will fight the pathogen. We have the dichotomy of the active immunity, which is basically natural and artificial. For the natural case, is basically catching a disease. So you would basically undergo the primary response and will produce basically your memory cells and antibodies respectively. But we also have the artificial thing, which is basically our vaccine. Vaccine is basically a harmless dose of antigen, which will also trigger humoral response. Therefore, produce antibodies and the memory cells for long-term perspective. We can also have the passive immunity. It is when we get antibodies only, in big amounts, so sufficient to fight the pathogen very fast. But make sure that you know that this type of immunity does not really produce the memory cells, which means that it will not really last longer than the antibodies will be present in the blood. Passive immunity is basically a given antibodies that were made by a different organism, so no memory cell. Just like in the active immunity we have natural and artificial. Natural is when, for example, baby is becoming immune to the disease as antibodies are transferred from the mother through the placenta or in the breast milk. And we also have the artificial passive immunity, which is basically getting injected with a large dose of antibodies, which are specific to the specific antigen. Okay, so now we are going to move to the past paper questions from International Pearson Edexcel biology course. Once you will see the question, please stop the video because the answers will show up very quickly. Thank you for watching.